Welcome to the Depth Dive. Today, we are getting right down to the fundamentals of biology. We're looking at our cells.
That's right. How they get injured, why they sometimes die, and importantly, how they fight back and adapt when things get tough.
Yeah, it's really the basis of, well, almost all of medicine when you think about it.
Absolutely. Understanding cell injury isn't just for researchers. It underpins pretty much every major human disease, heart attacks, strokes, cancer, even just the process of aging.
So our goal here is to kind of unpack the huge amount of information in the sources, looking at that whole spectrum, from reversible stress all the way to, well, cell death.
Mhm.
And there are two very different ways that happens.
Necrosis and apoptosis. We'll definitely get into those. The aim is to structure this, focus on the core ideas, the mechanisms that really matter.
Okay, let's unpack this then. Where do we start? What actually causes all this cellular stress in the first place? The etiology,
Right, the causes. The sources list quite a few, but you can group them. First, you've got the obvious physical things.
like trauma, getting hit, or exactly, trauma, but also extremes of temperature, hot or cold, and radiation, physical agents.
Okay. What else?
Then there are chemical agents and toxins. Think poisons, pollutants, industrial chemicals, but also things inside the body, like too much glucose or salt if levels get seriously out of whack. Even therapeutic drugs can be toxic at the wrong dose.
And nutritional issues.
Yep, nutritional imbalances, not getting enough essential vitamins, or conversely, excesses like obesity, both stress cells.
Makes sense. And I see infections are on the list too.
Of course. Bacteria, viruses, fungi, parasites, they all injure cells in various ways, either directly or through the toxins they produce.
Okay, so physical, chemical, nutritional, infectious. What about the really common ones? The sources highlight oxygen deprivation, right?
Definitely. Hypoxia, which is low oxygen, and ischemia, which is reduced blood flow. Ischemia is probably the most common and clinically significant cause.
Because no blood flow means no oxygen and no fuel delivery, and waste products build up. Precisely. It hits the cell from multiple angles. It's the root cause of things like heart attacks and most strokes.
Got it. And the list also mentions immunologic reactions and genetic problems.
Right. Sometimes our own immune system messes up and attacks our cells. That's autoimmune disease. And genetic abnormalities, well, they can cripple essential cellular functions from the get-go, like in inherited metabolic disorders or predisposing you to cancer.
So, a huge range of potential threats. But the cell doesn't just keel over immediately, does it? There's this idea of reversible injury.
That's crucial. If the stress isn't too severe or doesn't last too long, the cell can often recover. It can sort of bounce back if you remove the injurious stimulus.
And we can actually see signs of this reversible stress.
Yeah, morphologically, there are two main hallmarks mentioned. The first is cellular swelling, sometimes called hydropic change.
Okay, what's happening there?
Well, think about energy. Most cell injury impairs ATP production, the cell's energy currency. One of the first things that needs a lot of ATP is the sodium-potassium pump in the cell membrane.
Ah, the pump that keeps sodium out and potassium in, maintaining the balance.
Exactly. If that pump fails due to lack of energy, sodium starts to leak into the cell, and water follows sodium by osmosis.
So the cell literally swells up with water.
Right. You see these small, clear vacuoles appearing inside.
Yeah.
But if the energy supply is restored, the pump kicks back in, the water gets pumped out, and the cell can return to normal. That's reversible.
And the second sign?
Fatty change. This is where you see lipid vacuoles, basically little droplets of fat, accumulating within the cytoplasm.
Why does that happen?
It's particularly common in cells involved in fat metabolism, like liver cells or heart muscle. Injury can disrupt the normal processing and export of fats, so they just build up inside. Again, potentially reversible.
Okay, so swelling and fatty change mean the cell is stressed, but maybe okay. But what happens when the damage is too much, when it goes past that point of no return?
Then you hit irreversible injury, and the cell is committed to die. And this is where we see those two fundamentally different paths, necrosis and apoptosis.
Let's tackle necrosis first. You called it the messy one.
Yeah, think of it as accidental death, uncontrolled. The damage is so severe, usually involving things like ischemia or toxins, that the cell membranes just fall apart. Everything breaks down.
So loss of membrane integrity is key. The cell essentially bursts.
Pretty much. It swells massively, the organelles break down, and then enzymes, which are normally locked away safely inside lysosomes, get released.
And these enzymes start digesting the cell itself.
Correct. That's called autolysis self-digestion. And because the cell membrane is ruptured, all this internal debris, all the damaged molecules and enzymes, spill out into the surrounding tissue.
And that triggers inflammation.
Always. That's the defining consequence of the necrosis.
Yeah.
The leakage of cellular contents acts like a massive alarm signal to the immune system.
I
White blood cells rush in to clean up the mess, releasing their own chemicals.
Which can actually cause more damage to nearby healthy tissue.
Exactly. Inflammation is a double-edged sword, necessary for cleanup, but often causes collateral damage.
The sources mention different patterns of necrosis depending on how the tissue looks afterwards.
Right. Pathologists can often tell something about the cause based on the appearance. The most common is probably coagulative necrosis.
What does that look like?
Here, the underlying tissue architecture is actually preserved for several days. The cells are dead, they lose their nuclei, but the overall shape of the tissue remains intact, kind of like it's been cooked. The proteins denature and coagulate before the enzymes can totally dissolve everything.
And where you typically see that?
It's characteristic of ischemia, lack of blood flow in solid organs. Think heart attack, kidney infarct. Anywhere except the brain, usually.
Okay, so why not the brain? What happens there?
The brain typically undergoes liquefactive necrosis. Here, the enzymatic digestion completely dominates. The dead cells are totally digested, leaving behind this viscous liquid mass.
Like pus.
It's exactly like pieces, especially if it's caused by a bacterial infection, where you get a massive influx of white blood cells releasing enzymes. But you also see it in the brain after a stroke, due to the high lipid content and perhaps different enzymatic activity there.
Interesting. And the third main type was caseous.
Caseous necrosis. This one looks, well, caseous means cheese-like. It's typically found in tuberculosis infections.
Cheese-like, so white and crumbly.
Yeah, yellowish white, soft, granular, and friable. Unlike coagulative, the tissue structure is completely obliterated. It's often seen inside a granuloma, which is the body's attempt to wall off the infection.
Okay, so necrosis. Messy, accidental, inflammatory. What about the other side? Apoptosis.
Apoptosis is completely different. It's often called programmed cell death or cellular suicide. It's a highly regulated active process.
So the cell decides to die.
In essence, yes.
Yeah.
It activates a specific pathway that leads to its own clean dismantling. It requires energy, ATP, unlike the passive breakdown in necrosis.
And what's the mechanism? You mentioned caspases.
Caspases are key. They are a family of protease enzymes, sort of the molecular executioners. They get activated in a cascade, and then they go to work, chopping up the cell's proteins and DNA in a controlled way.
What does the cell look like during apoptosis?
It shrinks, the chromatin condenses, and crucially, the cell membrane remains intact, but starts blebbing. Then the cell breaks up into these neat little membrane-bound fragments called apoptotic bodies.
And these bodies contain all the cellular bits and pieces.
Yes, packaged up neatly. And these apoptotic bodies are rapidly gobbled up by neighboring cells, or phagocytes, the body's cleanup crew.
Ah, so because the membrane stays intact and everything is cleaned up quickly.
There's no leakage of damaging contents and therefore no inflammation. That's a critical difference from necrosis.
No.
Apoptosis is designed to remove cells without disturbing the neighbors.
And why would a cell need to do this? Why commit suicide?
Many reasons. It's essential during embryonic development to sculpt tissues. It's needed for normal tissue turnover, getting rid of old or worn-out cells. And critically, it's a defense mechanism to eliminate cells that are potentially dangerous.
Like cells with damaged DNA that could become cancerous.
Exactly. Or cells infected with viruses. Apoptosis gets rid of them before they cause bigger problems.
And how is this process actually triggered? The sources mention two main pathways.
Yes, the intrinsic and the extrinsic pathways. The intrinsic or mitochondrial pathway seems to be the most common.
Triggered from inside the cell.
Right. Triggered by internal stress signals, like DNA damage that can't be repaired, or a buildup of misfolded proteins, or even lack of growth factors. These stresses cause the mitochondria, the cell's powerhouses, to become leaky.
Leaky mitochondria, what leaks out?
A key protein called cytochrome C. When it leaks out of the mitochondria into the cytoplasm, it acts as a major signal to activate the caspase cascade, basically the powerhouse signals that things are beyond repair.
Okay, so that's the internal trigger. What about the external one?
That's the extrinsic, or death receptor pathway. Here, the signal comes from outside the cell. Specific molecules, often produced by immune cells like T lymphocytes, bind to death receptors on the surface of the target cell.
Like the TNF receptor.
Exactly. TNF receptor family members are classic examples. Binding of the ligand to the receptor directly activates caspases inside the target cell, essentially ordering it to die.
So internal sensors or external orders can both lead to this clean, caspase-driven death.
Correct. And it's worth mentioning the sources briefly touch on other pathways too. There's necroptosis, which is kind of a hybrid. It's programmed like apoptosis, but results in cell lysis and inflammation like necrosis, and autophagy.
Autophagy,
Yeah.
self-eating.
Yeah, it's fascinating. When a cell is starved or stressed, it can start digesting its own components, old organelles, chunks of cytoplasm, and membrane-bound vesicles called autophagosomes.
To recycle materials for energy.
Exactly. It's primarily a survival mechanism, trying to keep the cell alive during tough times by burning the furniture, so to speak. But if the stress is too prolonged or severe, autophagy can also contribute to triggering cell death, often apoptosis.
So a survival mechanism that can tip over into death. Okay, we've seen the outcomes, messy necrosis, clean apoptosis. Let's dig into how the initial injury actually causes this cascade. What goes wrong biochemically? You mentioned energy.
ATP depletion is central, especially in hypoxia and ischemia. Without oxygen, the main ATP-generating pathway, oxidative phosphorylation in the mitochondria, shuts down very quickly.
And ATP runs everything.
Pretty much all the essential housekeeping. Maintaining those ion pumps, synthesizing proteins, repairing damage. When ATP levels plummet, multiple systems fail.
We already mentioned the Na plus K plus pump failing, causing swelling. What else goes wrong?
Well, the cell switches to anaerobic glycolysis to try and make some ATP, but that's inefficient and produces lactic acid, lowering the pH, which messes up enzyme function. Protein synthesis drops off. And eventually, if ATP isn't restored, the membrane damage becomes irreversible, leading straight to necrosis.
So the energy crisis is a major driver. What about those free radicals we hear about? Oxidative stress.
That's another huge mechanism of damage.
Yeah.
Reactive oxygen species, ROS, or free radicals, are highly reactive molecules with unpaired electrons. Things like superoxide, hydrogen peroxide, hydroxyl radicals.
Where do they come from?
They're actually produced normally in small amounts during mitochondrial respiration, but cells have antioxidant systems to mop them up. The problem arises when production overwhelms the defenses.
Which happens during injury.
Yes, especially things like inflammation, radiation, chemical injury, and crucially, reperfusion injury.
We mentioned that earlier. When blood flow returns after ischemia.
Exactly. The sudden rush of oxygen back into damaged tissues can paradoxically lead to a massive burst of ROS production by damaged mitochondria and inflammatory cells. This oxidative stress can cause way more damage than the initial ischemia itself.
And what do these ROS actually do to the cell?
They attack everything. They cause lipid peroxidation, damaging cell membranes and making them leaky. They damage proteins, causing them to misfold or lose function. And they damage DNA, causing mutations or breaks. Just widespread molecular mayhem.
So the cell has defenses, antioxidants?
Yes, things like superoxide dismutase, SOD, catalase, glutathione peroxidase. These enzymes try to neutralize ROS. But in severe injury, these systems can be completely overwhelmed.
Okay, ATP loss, oxidative stress. Any other major players in the damage cascade?
The endoplasmic reticulum, the ER, is critical too. It's where proteins are folded and modified. If misfolded proteins start to accumulate due to genetic mutations, stress, viral infections, it triggers something called the unfolded protein response, UPR.
What does the UPR do?
It's initially adaptive. It tries to cope by slowing down protein synthesis overall and increasing the production of chaperone proteins that help refold the bad ones.
Trying to fix the problem.
Right, but if the ER stress is too severe or prolonged and the cell just can't clear the backlog of misfolded proteins, the UPR shifts gears. It activates pathways that trigger apoptosis.
So the ER can basically signal, this factory is broken beyond repair, shut it down.
That's a good way to put it. It becomes a pro-death signal when adaptation fails.
Fascinating. So cells aren't just passive victims. They try to adapt. Let's talk about those adaptations to non-lethal stress.
Absolutely. When cells face persistent but not immediately deadly stress, they can change their size, number, or even their type to cope better. These are generally reversible if the stress is removed.
Okay, let's start with changes in size or number. What's the difference between hypertrophy and hyperplasia?
Hypertrophy is an increase in the size of existing cells. The number of cells stays the same, but each individual cell gets bigger.
Like muscles growing with exercise.
That's the classic physiological example. Pathologically, you see it in the heart muscle when it has to pump against high blood pressure for a long time. The cells enlarge to generate more force. Or the uterus during pregnancy, that's physiological hypertrophy too.
Got it. So hyperplasia is different.
Yes, hyperplasia is an increase in the number of cells. It happens in tissues capable of cell division.
Examples.
Physiologically, think of the breast tissue enlarging during puberty or pregnancy due to hormones. Pathologically, Common examples include benign prostatic hyperplasia, BPH, in older men, or the thickening of the uterine lining, endometrial hyperplasia due to hormonal imbalance.
And the sources mention hyperplasia can sometimes be risky.
Yeah, while it's controlled proliferation, unlike cancer, it does create a larger population of cells, and sometimes the stimulus driving the hyperplasia can also increase the risk of mutations, creating, a fertile ground where cancer might eventually develop.
Okay, so hypertrophy is bigger cells, hyperplasia is more cells. What about shrinking, atrophy?
Atrophy is a decrease in cell size. This happens when cells face decreased workload, loss of nerve supply, reduced blood flow, inadequate nutrition, or loss of hormonal stimulation.
Like a muscle wasting away in a cast.
Exactly. Or the brain shrinking gradually with age. The cell reduces its metabolic needs to match the lower demand, often by breaking down its own components via autophagy, actually.
Makes sense. And the last adaptation mentioned is quite different, metaplasia, changing cell type.
Yeah, this is really interesting. Metaplasia is a reversible change, where one differentiated cell type is replaced by another differentiated cell type, one that's better suited to handle the stress.
What's the classic example?
The airways of a chronic smoker. The normal ciliated columnar epithelial cells, which are delicate, get replaced by tougher, stratified squamous epithelial cells like those found in the skin.
So the tissue adapts to withstand the irritation from smoke better. Seems like a good thing.
It's a trade-off. The squamous cells are hardier, yes, but you lose the normal protective functions of the original cells, like mucus secretion and the cilia that sweep debris away.
And there's a downside.
A significant one. Those metaplastic squamous cells, while adapted, are less stable. The factors driving metaplasia can also promote genetic instability, so metaplasia is often seen as a predisposing condition for malignant transformation. For cancer, the squamous cells in the smoker's airway are more likely to become cancerous than the original columnar cells were.
Wow. So an adaptation for survival can inadvertently increase cancer risk. That really links these concepts. Okay, wrapping up the adaptation idea, we have to touch on the ultimate unavoidable stress, cellular aging.
Right, aging isn't really an adaptation in the same way, but it's the consequence of accumulated stress and damage over a lifetime, coupled with a decline in the cell's ability to respond and repair.
What's the core mechanism driving this decline, according to the sources?
A major factor highlighted is telomere shortening. Telomeres are the protective caps at the ends of our chromosomes.
Like the plastic tips on shoelaces.
Exactly. They protect the important coding DNA from degradation during replication. But in most normal human somatic cells, a little bit of the telomere is lost with each cell division.
So they get shorter and shorter over time.
Yes. And eventually, they become critically short. This triggers a DNA damage response, and the cell enters a state of permanent growth arrest called replicative senescence. It stops dividing.
So there's a built-in limit to how many times most cells can divide, a biological clock?
Pretty much. This replicative senescence is thought to be a protective mechanism against uncontrolled proliferation, i.e. cancer. But the accumulation of these senescent cells, along with accumulated DNA damage and oxidative stress throughout life, contributes to the overall decline in tissue function that we see as aging.
It's the sum total of all these stresses and the waning ability to cope.
That's a good way to summarize it. Aging is that progressive decline resulting from all these factors converging.
Okay, so pulling this all together, we've seen cells under attack from all angles. We've seen their immediate responses swelling, maybe recovering, and then the stark choice between messy necrosis or programmed apoptosis if things go wrong. And we've seen how they try to adapt long-term, sometimes successfully, sometimes with risky consequences.
Right. The key takeaway is that it's a dynamic process, a spectrum. The same injury, like ischemia, doesn't just cause one thing. Depending on how bad it is and how long it lasts,